Hi all, Dr. Clark here again. Today we're going to talk about the second piece of Hardy-Weinberg, in that we're going to talk about ways which either natural, artificial, sexual, something's going on that can violate Hardy-Weinberg equilibrium. So in other words, we're going to talk about evidence for evolution, or means by which evolution can occur. Okay, classic examples of population shifts, like the peppered moth example, where due to industrial revolution you have what we call industrial melanism, and so species that might have been suited for an environment where there wasn't a bunch of soot, dark background, and it was more light background, they get replaced by individuals that have just black, in this case wings, of a moth, the peppered moth. But other examples include lizards, and there's other species of insects that we see the shift to due to industrial melanism. And then once we clean up the environment, we see the shift go back. Okay, but really what's happening is your population is shifting, and with that shift, what we're talking about is evolution of the population. It's moving from one extreme to the other, and you get this big shift in population where, yes, there might be some individuals that are peppered still with the white and black, and that's represented here, but they only represent maybe 10% of the population, where 90% of the population is the dark form, and then you get the switch back where 10% might be the dark form and 90% the white and black form. And we'll talk about these different examples as we progress. But remember, in all these cases, we're talking about violating Hardy-Weinberg equilibrium. A violation of Hardy-Weinberg equilibrium means that the allele frequency in the population is changing, and therefore evolution is occurring. Okay, so ways in which this can occur. One that occurs with endangered species often, or at least species with very small population sizes. So if you remember Hardy-Weinberg, the very first rule for population to fit Hardy-Weinberg equilibrium is their population needs to be large. In this case, if you have small populations, you get something called genetic drift, or what sometimes is called neutral evolution, meaning that it's not really predictable, and it can swing. The pendulum can swing in one direction very quickly, and in some cases, if the population's small enough, it can swing completely over and eliminate alleles. Okay, so you can get fixation of all recessive alleles or fixation of all dominant alleles, and you have no diversity in that genotype or that gene. Okay, and so these are chance events. Right, this is just by chance the frequency of genes of the population is shifted. It changes. Okay? Now you can think of this as like a coin flip. If you take a fair coin, a quarter, heads, tails, you would expect, if you flipped it 10 times, for you to get 5 heads and 5 tails. Now, that's the probability. But because you're flipping it only 10 times, the chance of a freak event, or the chance of a anomaly to the 5 and 5 is much greater than if you flip it 100 times. You can test this out. So flip a quarter 10 times. Okay, you would expect it 5 and 5. Probably not going to get that, because just by chance you're going to get more heads or more tails than the other. Then flip a quarter 100 times. You'd still expect it to be 50-50, okay? So 50 heads, 50 tails. And I would probably be willing to bet that you're going to be fairly close to that. Maybe 49, 51, somewhere like that. And I bet you if you look at the actual range, you're closer with 100 flips to 50-50 than with 10 flips to 50-50. Even though the probability is identical, the chance of an anomaly, or the accidental portion is going to be greater on 10 flips than 100 flips. And this is what we see with small populations. When you have really small populations of organisms, the chance that one male fertilizes all the females, depending on the species, is much greater than if your population is much larger. Okay? Or the chance that one male doesn't mate that has this rare allele because maybe, it wanders off and is finding food, or maybe, it gets, into a region where the habitat's not good and it can't find a mate, something like that, and the mating season is really short. Maybe the male with a rare allele doesn't mate that year. Okay? So you can shift your genetic drift, or you can shift the genes of the population in one direction. Okay? So this is probably most important when we talk about random fertilization events, because if genetic drift occurs most often when things are random, like there's no mate choice. You can really get this shift when it's just whichever male's ready, when the females is ready, is who's going to fertilize. So this happens a lot in fish, where all the males, they're starting to mature and the females mature. The males that are ready are the ones that are going to get to lay their eggs. So the males that are moving into the region are the ones that are going to get to fertilize their eggs, not lay their eggs, fertilize their eggs. Okay? So those are kind of just random fertilizations. Now, if you get a bunch of small males that come in and they're the ones that fertilize, then the gene or series of genes for decreased growth rate or small stature, they're going to be more prevalent in the population. You're going to get a shift. All right. So there's a couple other ways at which genetic drift can occur. You can get this naturally by just having small populations, or it can be driven by specific events, like the founder effect. Okay? So the founder effect is when a few individuals from a population migrate, move, get transplanted, something, into a new ecosystem, a new environment. Okay, so you're not selecting for certain traits, you're just pulling a few individuals, they take their genes with them, and they get established in a new region, and that's the gene pool. So you can get a huge shift in gene pool from the original gene pool of the original population. This happened in Pennsylvania Dunkers. Okay, they migrated to a portion of Pennsylvania. They only mate within their community. They don't mate outside. No one can come into the community and seek mates and reproduce. So you'll see that they have a lot of genes that are very specific to this group of people. Some people would call them inbred. Okay, but even at that, they do have some alleles that are fixed in the population, and some of them are maladaptive, and some of them have no real effect. Okay, but again, we can see that they're very genetically distinct compared to other portions of people in Pennsylvania or across the United States, for that matter. The other way to do this is by creating a bottleneck, okay, or what we call the bottleneck effect, which means that you take a large population, and then through some means, that population is shrink a lot. Okay? Maybe drastically. Maybe you go to 10% of their original population. Okay? Well, by taking that out, you take a lot of the diversity, the genetic diversity, and the gene pool is removed. Then the population is allowed to rebound. And so you go from big to small, back to big, but the result is you don't have very much genetic diversity now compared to what you used to have, and so you shifted the gene pool in one direction. And so it's a result of evolution, okay, but it's typically not very adaptive. Take cheetahs, for example. Okay? Cheetahs were bottlenecked due to hunting and over-harvesting. So they used to have a huge diversity of alleles, 25 different alleles in a region. Okay? After the bottleneck event, only 4 remained. And so now when we look at cheetahs in certain regions, they are nearly genetically identical. There's only 4 alleles left in the population. Okay? This can be very harmful for an organism because what if some of these alleles, some of the 25, were adapted to climate change? We're adapted to an immune system. We're adapted to resistance to certain diseases, bacterial infections, fungal infections. Yeah? These kind of things are gone now from the population. So if a disease comes through that you used to have resistance to, now it could wipe out the entire population, and the entire population could go extinct. The other thing is, we've done this with multiple species, things that we've hunted to near extinction numerous times. Cheetahs, northern elephant seals, fur seals, canvasback ducks. I mean, tons and tons of species have been hunted. Bison have been hunted to near extinction and then brought back from the brink of extinction, which is great, okay? Because that's the purpose of the Endangered Species Act. But in the end, really what you've done is you've removed many genes and many allele differences in the gene pool, and now you're stuck with a very small sample, and it can be very harmful for the organisms. So elephant seals also went through this bottleneck. Elephant seals, these big organisms, they'd never seen humans before, and humans would go to the region and they wouldn't move, so humans would just club them. And they would take their fat for oil for lamps. Sometimes they'd take their skin and things like that, but most of the time it was just fat, so they just take the fat and that's it. And really, the population was decimated. The best estimates is that the population got down to a little over 20 individuals on one small island. And then in Mexico, the Mexican government started protecting some of the islands that these organisms used to reside on, and their populations have started to rebound. And now their populations are pretty large, and maybe 20, now they're probably somewhere in the tens of thousands. But you started with 20 organisms, and so genetic diversity is very low, and so there's a lot of concern with diseases and things like that, that these organisms might be subjected to some kind of bacterial infection or fungal infection, and it'll wipe out the northern elephant seal from the planet. Right? All right, so gene flow. Gene flow is another method in which we can get a change in allele frequency over time, and that's normally through migration events. Okay, so that's what gene flow means that genetic material is moving from one region to another. Okay, now yes, founder effect can both be genetic drift and gene flow. Organisms could migrate to a new region that they've never been, taking material with them. Okay, that's genetic drift, but gene flow could be the opposite. They could be taking distinct genes out of the original population. So you could have both at the same time. And we'll talk more about evolution because we often think about evolution from a just kind of a generalized perspective, like, oh, that's natural selection, or oh, that's sexual selection. Okay. But in actuality, maybe you do have natural selecting acting upon some genes, while sexual selection is acting upon a few other genes, and you got gene flow acting on some genes, and you get some mutated genes because they're less conserved, and so you get this mosaic of genetic variability and gene I guess you could call it gene jockeying. Okay, so the population is multiple genes. Okay, and some genes are under the influence of sexual selection, natural selection, different things. Okay, so back to gene flow. Gene flow can work in a couple different directions. You could have genetic material moving out of the population and into a new population. So one population could be getting less diverse, while the other population could be getting more. And so it really just depends on how big the populations are, how genetic diverse the population is, and then what organisms are actually leaving the population. Are they genetically unique, or are they, average, like normal? Okay, so they're in the middle, they're intermediate from most, for the most part of their genes, so it's really not altering the, the genetic frequency in that population. Here's a good example of a lack of gene flow. Okay? Most people don't know this, and I often talk about elephants when I talk about the absence of evidence for evolution, because elephants don't reproduce very fast. Okay? Elephants normally wait probably 20-25 years before they become sexually mature, depending on the species, okay, or subspecies. And then they only have a calf maybe once every 5 to 10 years. Okay, so we're talking in an elephant's lifetime, even if they're super reproductively active, they might give birth to 4, 5 calves in their entire life. So 4 or 5 calves in their entire life. So to get evolutionary change, it takes quite a bit of time for that to occur. Even at that, because we as humans have isolated populations and we've stopped gene flow between populations, we have what we call a savanna subspecies of elephant and a tropical forest subspecies of elephant. And you can tell the difference between these. And most people could tell them if they were standing next to each other, you could tell the difference. The savanna is much larger and has white tusks. Okay? Along with that, there is some a little bit of difference in kind of ear shape and things like that, that if you knew what you were looking at, you could tell, and a little bit difference in the mass of the head, and you could tell, and the age at sexual maturation. The tropical forest subspecies is smaller. Its tusks tend to be more red. Now, part of that has to do with them scraping the tree bark and things like that. But these guys have white tusks due to kind of the sun bleaching. So you can tell the difference between the two, but really that's probably not genetically controlled. Size is genetically controlled. The head features are genetically controlled. But the idea is these two populations used to mix. So you used to have savanna elephants that would move into the forested habitat during different times of the year. And you have the forested elephants, would be moving in the savanna. It was one big population. They'd move back and forth together. Well, through, either hunting pressure, habitat destruction, something, now we have forested elephants that don't leave and savanna elephants that don't leave, and they're starting to diversify. And in many cases, most mammal biologists believe that these are actually not subspecies, but these are two separate species now. Okay? And that's due to a lack of gene flow. Okay? There is no mixing between the populations, which we call isolation or reproductive isolation. All right, mutation. Mutations kind of get a bad rap just because, everything that we read and we're taught when we're young is that mutations are bad. They cause maladaptation, weird things to occur, things like that. But mutations are really the source of all new alleles. So even though majority of mutations, it's true, the majority of mutations are either maladaptive, meaning they cause harm to the individual, or they're neutral, they don't do anything. Okay? There's very few mutations that pop up in a population that are beneficial mutations. Now when they do, and they're beneficial, then they're quickly acted upon by natural selection. And so the individual will reproduce more offspring with that mutation, and their individual more offspring with that mutation, on and on and on, and you get this big shift and a quick adaptation to new environment through natural selection, or in some cases it's not natural selection, but sexual selection. So you might have a mutation that the female of the species is attracted to, and she picks that individual all the time, and then their offsprings are picked by other females, on and on and on. Okay, so the origin of all new alleles come from mutations. Okay? Again, mutations are not severe enough in most cases to change the allele frequency of a population to drive evolution. However, with mutations, coupled with sexual selection, artificial selection, natural selection, you can get a change in the population's genetics. Okay? Mutations are random for the most part. Now, we have some mutations in certain regions of our genome, humans and mice and other genomes that we know of, that we have determined a rate of mutation. Okay? Now, it doesn't mean that it's exact, okay, but it's give or take a few hundred years or so. It depends on the mutation. But we have a range at which mutations will pop on certain genes. Okay? These are genes that will be less conserved and have more ability to change. Okay? Like I said before, most mutations are detrimental or neutral. Okay? And the only ones that are getting passed on to the next generation are the genes that are affecting the gametes. Okay? So they are the mutations that are affecting the gametes. So these mutations have to occur in sperm or egg, or the germline cells that give rise to sperm and egg. That's the only way that a mutation is going to be passed on. Like I said before, even though they're random, even though they're not super predictable, there are different levels of pressure on certain genes. Some genes, they're just not going to mutate. Okay? A mutation will cause the individual to die. Okay? And so you don't have very many mutations in that region. It's very rare. In other regions of the organism, mutations happen much more often because the organism can live with that mutation. Now, it might not be adaptive, it might not be, good for the organism, but they can live. So, for example, like hair placement, hair color, hair texture in humans, right, can pretty much live without hair. So any adaptation to the hair or any mutation to the hair doesn't really make a difference in modern humans. Okay? Now, other organisms can't really mess with some of those things. Like, you can't remove all the feathers from a bird through a mutation. And for that matter, you can't really change the flight feathers. Otherwise, they won't be able to fly correctly. Okay? So there are some genes that have high pressure, okay, which means they're not going to change very often. And then other genes that the pressure to mutate is kind of low. In other words, there's nothing stopping them from mutating. And on top of that, the checkpoint system that we have might not check that region as often as it would other regions. Okay? We'll talk more about mutations later. All right, so natural selection. Natural selection kind of works in three separate ways. Okay? You can have what we call directional selection, where one extreme is selected for and one is selected against. So you get this shift in one direction. Okay? Disruptive selection, where both extremes are selected for, intermediates are selected against. So eventually what happens is you kind of get the extremes on both ends and nothing in between. Disruptive selection, if carried on long enough, will typically result in speciation. Okay? Because eventually the extremes won't recognize each other and they won't mate. Okay? And then you have stabilizing selection, where the extremes are selected against and the intermediates are the ones that prevail. Okay? So we'll look at some examples. Again, back to the peppered moth example and industrial melanism. Okay? So prior to the 1950s and the Industrial Revolution, we had this form, this peppered form, white with black spots or gray spots. And it fits this background pretty good. Okay? So they have paper birch in Europe. So most of this data is coming from Europe. And so the peppered moth had has this coloration that fits the paper birch trees, okay, very well. So it's harder to pick them out. There's a moth right there, okay. And then there's dark form, melanized form, okay, which would be black or dark gray, and doesn't fit the light tree very well, but definitely fits the soot-covered tree very well. Okay? And so what we see is we see this population shift, because in one case, a phenotype is a disadvantage, and in the other case, it's an advantage. And so you get this shift. And what we saw was a shift during the periods of what we call industrial revolution, where there was no check on pollution and carbon dioxide and these kind of things, to a period where the Clean Air Act came in the 70s, and you started to see that the shift moved back. There's less melanized black, gray moths in the population as these trees started to become light, and there was less soot, and the new trees were growing, and they weren't soot covered and things like that. The peppered moth, the original version, got to come back. So it went from like 90% melanized during the Industrial Revolution, the heyday of it, to 90% white with black specks today, okay? And you still got 10% here and 10% there, respectively. Okay? All right, disruptive selection. Again, remember that this is when we're talking about species that are selecting, or systems that are selecting for the extremes of a population. So the midshipman, which is this fish here, okay, this is a female. This is a large male that holds territories. This is a small male, which we call a sneaker male, and they kind of look like females, or at least juveniles. Okay, so large males will defend territories. They'll mate with females. They don't allow any other males to come in, no intermediate males, no other large males. They fight them off, etc. But these little males right here, which are reproductively active, they do have the ability to produce sperm. They do have the ability to fertilize eggs. These are called sneaker males or miniature males. They'll come in. This male doesn't react to this other male, either because it thinks it's a juvenile or it thinks it's a female. This male will fertilize some of this female's eggs. Okay? And so you get these intermediate phenotypes, those medium-sized individuals that are selected against. If you're not big enough to hold the territory, but you're not small enough to sneak in, then you're going to be out reproduced, and there's no reason for you to occur in the population. So we have two subpopulations of males when we're talking about midshipmen. And like I said before, eventually if these sneaker males start mating with small females, and the large males start mating with just large females, then you might get this big shift, and you might get two separate species due to reproductive isolation. All right, sexual selection. So when we're talking about sexual selection, we need to talk about situations where, and this will be evident in class, we'll talk about structures that just don't make sense. Like, having birds that are red and singing at top of the tree while the female is brown and sitting at the base of the tree and well hidden, that doesn't make sense from a natural selection point of view. Why would a male present himself to predators and to females, okay, and sit there and wait for a predator to eat it? Okay, well, it's because females are driving that. Females want males that are brightly colored. They want males that can sing. Okay, we're talking about birds, but this is an example of fish. There's an example from this of like large mammals with big antlers, okay, or large tusks, these kind of things. Females are selecting for individuals that have, unique adaptations, okay, in some situations. That's sexual selection. A lot of those adaptations are not beneficial when it comes to natural selection. Okay? So you get these extreme morphologies sometimes. Okay? And we can get situations where, you know, tail feathers are very different, okay? Or you know, get bright colors in the sense of, you know, fish or amphibians, or, you know, birds. You can get large antlers, tusks, horns, all kinds of things that are going on. Now, one of the key things that I want to talk about is back when I said that some genes are conserved and some genes are not. Okay? So you can alter probably the tail feathers of an organism to a degree. Okay? Now, peacocks are an extreme. Peacocks have tail feathers so large that it makes it very difficult for them to fly, and in many cases, they don't fly because it's just the capability is pretty much gone. Versus something like, you know, a golden eagle, which will have different tail feathers and even tail feather coloration, but it's not to an extreme. It's not growing extreme tail feathers. Okay? Instead, you know, it might have different bar colors. It might even be different colored at the tip. You might have a different, you know, primary color in some of the subunits here in the tail. But overall, the color is pretty much the same between birds, but you can see slight differences in coloration. Okay? And if you knew what you're looking for, you could probably individualize birds based on the color of their tail feathers when it comes to things like golden eagles or really other birds that have kind of a modeling to their tail or to their breast for that matter. It doesn't really matter. Okay? All right, back to other selection methods through natural selection. Stabilizing selection. This is when the extremes are selected against. So a good example of this is horseshoe crabs. If a horseshoe crab, because it's an aquatic organism, but it has to lay its eggs on the terrestrial environment. Now remember, these organisms are breathing with gills. They have a series of what we call book gills underneath, okay, this region. And so when they move out of the water, they cannot breathe. They have to house water underneath the book gills and slowly take the oxygen out of that water, okay, in order for them to respire. Now, so they can't spend very much time outside the water. One thing is if you're really large, you take up a lot more heat while you're outside of the water. You have a lot higher evaporation rate, okay? And so large horseshoe crabs don't make it very well. The other extreme is being very small. If you're very small and you move on to a terrestrial habitat, you're going to be picked off by birds. Even with this hard carapace shell on top, birds will eat you if you're small. Okay? And for that matter, even in the ocean, you're going to be food for a lot of things. So the two extremes are selected against. One through thermal regulation, temperature profile, and the other one through predation. So you get these intermediates in between, and they have a fairly narrow phenotypic range. Now, one of the concerns is because one of the ranges, the top end, is based on thermal regulation, with the planet changing, okay, going through warming periods, what we're seeing is that this size range is actually kind of shrinking on horseshoe crabs. And so part of the upper end moving towards the intermediate even more could be because of those thermal regulatory behaviors or inabilities, okay, with global climate change, temperature increases. They have a very narrow phenotypic range. So often people will say, well, the planet's only increased by 1.5 degrees Celsius. Okay? Well, 1.5 degrees Celsius to an organism that has like a 5 degrees Celsius range in which it can expand. Okay? That means you're taking off 1.5 degrees or you're putting on 1.5 degrees, and so that organism cannot survive. So now it's got about a 3.5 degree range at which it can survive. So for some organisms, it's a really big deal for that shift to occur. All right, so species and speciation. We kind of already talked about this biological species concept. Remember, just means that Organisms that can reproduce and produce viable offspring are considered species. Okay, that's kind of an old species concept. There's lots of problems with it. First of all, if it's an asexual species, it doesn't really work. Most of the taxonomy is done through morphology, so physical characters, not whether or not things can mate. Any extinct species, very difficult to tell if they would have mated with each other. So most of that's done from morphology. So all fossils, I mean, you can't see if they mated or not. So it's really just, it's difficult to establish a true idea of what a species is using the biological species concept. So instead, what's been created and what's used during modern times is the phylogenetic species concept. And so this is, a lot of this includes biological species concepts. So it includes the genes, the genetics, the interbreeding capabilities, but it also examines the ancestral connection between species. Sometimes the phylogenetic species concept is based purely on genetics. Sometimes it's based on morphology, genetics, behavior, and breeding capabilities. And so the better the phylogenetic tree, the more variables that connect things and the more variables that make things clear and that are presented on the tree. And you'll see what I'm talking about in a little bit. So speciation, again, or the formation of new species, really comes with isolating subpopulations. So remember, the key to speciation is sexual isolation. And so if species cannot, subpopulations, cannot mate, they're isolated, then any mutation that pops up in one population, it's not necessarily going to pop up in the other, so they can be adaptive to a new natural environment. That mutation could be adaptive, and on and on and on. This environment might select against different traits than this environment, and eventually those two isolated populations can become new species. So how isolation works is there's quite a few different ways at which you can get reproductive isolation. Here I just kind of list a few. So you can have geographical barriers. This could be an island and a mainland and the ocean in between, and they can't traverse. It could be a river split in two things. It could be a volcano erupted, splits two things. It could be a mountain range. All kinds of things. Humans can do it with a 8-lane highway separating two populations. A city that separates northern population from southern population, and they're not migrating through the city. There's lots and lots of geographical barriers that can occur that isolate populations. Behavior attributes, okay? This could be different dances, different singing capabilities, different fighting, all kinds of behavioral attributes that can separate populations. A great example of one that's occurring currently today is house sparrows that occur in Europe. The ones that occur in urban environments sing a different frequency than the ones and amplitude than the ones that occur in rural environments. And it's so drastic that the rural environment birds cannot recognize the urban environment birds. And the urban environment birds, or the ones that live in cities, are singing louder and a different amplitude to combat the traffic noise and the normal city noise. So they've changed their song. So males change their song. So females can find the male and mate with the male and hear his song over the ambient noise. In rural environments, they never changed. So those two don't recognize each other anymore and eventually will become separate species. So those are some behavioral things. Reproductive physiology, this could be a couple different things. First of all, could be that the vaginal cavity is too acidic or too basic. The sperm don't have the capabilities to have the right binding, okay, which allows it to bind to the egg. It's a protein, or lysine, which is another protein which allows for it to enter the egg. Those could just be, a different protein shape, and it doesn't fit with the egg. So and there's lots of other reproductive physiologies that can go on that can alter subpopulations and not allow them to mate with each other. And then you can get hybrid non-viability, which means that the sperm and egg might fertilize each other, but because of the difference in the number of chromosomes or size of chromosomes, you can get maladaptive individuals, or you can get individuals that are born that are sterile. Okay, like a horse and a donkey producing a mule. Okay, sterile individuals due to different sizes of chromosomes and different chromosome makeup, and they don't produce viable offspring. There's lots of others. There's temporal isolation, so different times of the day. There seasonal isolation, different seasons. There's all kinds of isolation events that can separate two populations reproductively. All right, let's examine allopatric speciation. Okay, we already just talked about this. This is when populations are geographically isolated. Okay, so they're isolated from one another, and then genetic drift, natural selection, sexual selection can take part and act upon those isolated subpopulations independently. Okay? Parapatric speciation is kind of a unique one. This would be an event that occurs typically in very small local populations. Okay? It means that these populations are not reproductively isolated. They could reproduce with each other, but frogs that occur in, say, one pond and another pond, and they just don't make the migration. They can make the migration, but they don't. Right? They don't leave the pond, they don't migrate, and they don't reproduce. Sea urchins that live in particular tide pools could move to another tide pool and mate with another group of sea urchins, but they don't. Okay? Now, for the most part, I'm not entirely sure. I'm not super convinced that parapatric speciation occurs when people have demonstrated that it occurs. A lot of this might be, now this is just my opinion, a lot of this might be that you have a lack of evidence, or the study didn't go long enough. So instead of seeing something like sympatric speciation, or seeing that these organisms are actually mating with each other, the study was ended and it's deemed parapatric speciation. Okay? Even when it does occur, due to through scientific studies and it's been documented, it's extremely rare. Okay? The other one is sympatric speciation. Okay, so this is isolation within a single population. Okay, and this can just be a huge variety. It could be due to behavior. So one individual hatches in the morning, another group hatches in the evening, and they never encounter each other. Insects do this all the time, so they occur same population, same body of water, same soil samples, whatever, but because they hatch at different rates, they never encounter each other, they never mate with each other, and they're two isolated populations. Feeding in different regions of the water, so you can get fish that feed in benthic on the bottom, fish that feed in pelagic up near the surface. They never encounter each other, they don't mate with each other, therefore they're isolated, even though they occur in the exact same body of water. Okay, indigo birds, brood parasites. Okay, so there's quite a few brood parasites out there. The cuckoo bird, brown-headed cowbird. These birds lay their eggs in other birds' nests. Okay? But in order for it to be advantageous, it needs to be laid in that other bird's nest, and the offspring need to vocalize like the hatchlings of whatever the host bird is. So if you take an indigo bird and you drop it into, say, a widow bird's nest, it needs to know the vocalization of that widow bird. If it doesn't, it's probably going to be recognized, it's probably not going to get fed, so it'll die. So there are portions of indigo populations that they recognize that offspring is going to recognize a different host bird. Okay? And so you get this isolation. Maybe one recognizes, widow birds, one recognizes like blackbirds, one recognizes another group of birds, and so they're manipulating multiple birds' nests, multiple host birds' nests, but those populations are isolated because of that advantage. Okay? Other examples are things like different species of cats that hold territories. They'll often hold specific territories, exact spots where they will go and visit the regions of their territory, and they go to those different regions of their territory at exact times of day. They prefer specific types of food and habitat and things like that. So you can, even in a forested habitat, you can isolated populations where they don't encounter each other because they don't recognize the other activity pattern or the other individuals' territories or preferred foods or things like that. Same species, same population, but they're starting to become isolated because of differences in daily activity patterns or something like that. All right, rates of evolution. We have a couple rates of evolution I want to talk about. First one is phyletic gradualism. Okay? This is gradual evolutionary change over millions of years. Mammalian evolution, at least humans, this is an example, a pretty good example of this. We gradually changed, gradually adapted, going bipedal, okay, decreasing the jaw size, increasing the cranium, having the ability to speak, use tools, communicate, have societies, on and on and on. That's phyletic gradualism. It didn't happen all in one day. Small steps, years after years after years, millions of years of gradual evolution. Versus something that regard to punctuated equilibrium, where you have long periods of no evolution and then an explosion of speciation events. Okay? And so you can get like Galapagos finch is a good example of this. Anything that's moved to a new environment, new habitat, can experience this, because you'll get individuals that move to that region and then they occupy all the different ecological niches, or have the potential to occupy all the different ecological niches. Either they'll go extinct or you have an explosion of species. And that's exactly what happened with the finches. Came from, Ecuador, made it to the Galapagos Islands, and then started to go through adaptive radiation and just started to have many, many speciation events, okay, and lots of adaptations to the current environment. So here you can see the two. Okay, so you get rates of evolution if you got gradual change over a long period of time, okay, with diversification, one species giving rise to maybe a couple species, etc. Or you can get punctuated equilibrium, nothing for a long time, and then boom, rapid. And so you could have not just one species off here, but you could have like C, D, E, F, G, all kinds of species that are coming off after a long period of stasis. All right, molecular evolution is probably the best evidence that we have for evolution today. Okay, so Darwin, when he was talking about fossils and analogous and homologous and all these other characteristics, nothing compares to the evidence we have for evolution through genetic analysis. Okay, and so we know this as basically the central dogma theory. We know that if we alter a DNA sequence, we're going to alter the RNA sequence, which is going to alter the protein sequence. Okay? And so that's called the central dogma theory, that DNA is transcribed into RNA and then translated into proteins. Okay? And any changes in the DNA will alter the protein. By knowing that, we can also examine things that we consider conserved. Okay? These are genes that are in an organism that have not been changed in a long period of time, because any minute change will result in death of the organism. Okay? So things like cytochrome C in the mitochondrial genes, this doesn't change very often at all. Now it does show relationships, distant relationships, but cytochrome C hasn't been changed in mammals in millions of years. Okay? Same genes, same everything, same process, and that's because it occurs in the electron transport chain. It's a protein that's embedded in the mitochondrial matrix. Okay? It allows for protons to move freely across it, and without it, you have no ability to do the electron transport chain and convert, protons, a proton gradient into ATP. Okay? Without it, it's not possible. Okay? And so even when we look at bacteria, they have a cytochrome C. They don't have mitochondrial genes, but they have a cytochrome C gene protein that's embedded in their membrane and have the capability of using it for energy purposes. Now there are less conserved regions, which we've talked about before. Okay? These are going to accumulate more mutations in a shorter period of time, okay? And they often are very useful for establishing relationships of closely related organisms. Okay, so you got conserved genes, which we can examine and look at the big picture. So that we can look at the domains, we can look at eukarya and archaea and bacteria, and we can look at how the domains fit out, how the kingdoms fit out. But then if we're trying to figure out what's the difference between this species and this species, we want to look at the less conserved regions that have probably mutated in the last million years. And that will give us a relationship of how those organisms are related. Okay? So here's a good example of kind of conserved genes, cytochrome C. You can see that most people can distinguish a cow and a sheep. You know that they're not the same species, but there is no difference in cytochrome C genes between those two. The difference between cows and whales are 2, okay? And most people would definitely say that they could tell the difference between those. Horses and cows are 3. Now people would be like, well, why is a cow and whale so closely related compared to a horse and a cow? Well, the cow line, that ungulate line, gave rise to whales. So whales were once terrestrial, that line was once terrestrial, and then migrated into a marine environment. So they were probably half marine, half terrestrial, and then full marine. Okay? And so that's why there's that connection, and it's more closely related than horses and cows. Horses are not related at all to cows. They have a very different line. Horses being a single toe, they started out with 5 toes, and a very different line than the cow line. But on and on and on, you can see the differences there. These are very conserved genes, so even at the, some of the most diverse things, a yeast, which is a fungi, versus an animal, a moth, there's 44 differences, which is not all that many differences when we're talking at least probably 1,000 amino acids to make a cytochrome C protein, and only 44 of them are different. Okay? So one thing that I want to drive home is that species are a mosaic of evolutionary differences. Okay? And being a mosaic model, you have some genes that are conserved, don't change very often at all. Some genes that are less conserved, they change all the time. And some regions of the DNA that don't actually code for anything, that change constantly. Okay? And so from a point of view of mosaic evolution, not only do your genes change and differ based on the rate that they change, the ways that they can change differ. So they could change via natural selection, sexual selection, in some organisms, artificial selection. They could be the result of genetic drift, gene flow, lots of different things to create this mosaic. Okay? And so mosaic evolution is really the combination of conserved and less conserved genes. Okay? So you can examine things like birds, and you can say, well, highly conserved genes are things like having a bill, okay? Having wings, having feathers. Less conserved could be related to some of these. You could have feather color. Even how the wing forms, like the length of the feathers, can be a little bit less conserved. Okay? The leg formation and function, where the toes and the digits are how they face, how long they are. These are all very, very adaptable characteristics that the result is probably not going to cause the organism to die in the environment. Now, again, wing formation, if your wings are too short and you can't fly, then of course the organism is not going to make it. Okay? But less conserved genes often are the ones that differ between species. If you examine one species of sparrow and another species of sparrow, they might differ in color, they might differ in length of the wing, they might differ in where the toes point, how long the toes are, the leg formation, okay, these kind of things. But those are differences between those species. Okay? All right, so with that, we'll change and we're going to start talking about systematics, so how organisms are classified, and then we'll move into the precursors to the animal line, which we call protozoans.